Okay, so we're now going to hear about two new diseases, Leiomyosarcoma first from George Demetri, multiple myeloma from Nikhil Munshi, and we've highlighted the diseases because they are new, they're important, and there's set-aside funding in support of grants for these diseases, so pay close attention. George, take it away.
Well, the reason we are, first of all, thank you for allowing the leiomyosarcomas into this August group. This is a wonderful opportunity to build some new collaboration because sarcomas, being mesenchymal malignancies that are not a public health crisis, are nonetheless very interesting biologically. We've learned a lot from them. So let me just tell you what they are. They are cancers of things that hold the body together, as opposed to epithelial cancers, which usually are glandular. They serve some other physiologic function. These have a connective tissue function in general. As you remember, mesenchyme or embryonic connective tissue really develops into a variety of different tissues, including the real connective tissues that we consider like muscle, fat, bone, cartilage, etc. The blood vessels and lymphatics are in this. And then the blood cells of all lineages are also developed from mesenchyme. So mesenchyme is a very interesting set of tissues. Sarcomas can show up anywhere in the body. And most people say there's about 16 to 18,000 new cases of sarcomas of any type in a year in the United States. 1% of adult cancers, but 15% of pediatric cancers because the kids don't get the complex carcinogen-driven cancers like lung cancer and other things like that. So if you look at that, most cancers are carcinomas. You get that smattering of blood tumors, melanomas, and then sarcomas are the really sliver of the green there. Sarcomas are a really extreme element in heterogeneity. Even though they're relatively rare, they come in all sorts of different colors and sizes from standard histopathologic variances. And when you look at these, they're a really diverse collection. When you look molecularly, most of them, 18% anyways, the biggest fraction, are these gastrointestinal stromal tumors, where we've made a lot of headway because they're mutationally driven. We certainly know for greater than 95% of them, they're driven either by driver mutations in KIT or platelet-derived growth factor receptor alpha, and it's been the basis for really revolutionizing care through targeted kinase inhibition. So just as one type, we've learned a lot from that. It actually gave us rules that we could then play in the non-small cell lung field, as well as other targeted therapies that are mutationally activated. Leiomyosarcomas, or smooth muscle tumors, are about 16% of all sarcomas. The next most common are fat tumors, and then a smattering of other things, including 10% that are truly unclassifiable. Now, how does one diagnose a leiomyosarcoma? It's still based on von Leeuwenhoek technology from the 1600s, really. It's good pathologists with pattern recognition, looking at the stains, blue and red stains, looking at the unique morphologic criteria of these cells. What the bridge project that we're proposing is to move beyond this and really help us to hit this disease in a different way. When you look at these, the histopathology, as you can see, is just these elongated cells with so-called cigar-shaped nuclei, occasionally more pleomorphic nuclei, and that's really the gold standard, along with immunohistochemical staining to back that up. So desmin, smooth muscle actin are key immunohistochemical criteria for this. So what are these cells? We don't know what's driving them. There's no identified mutated or other drivers. There's a smattering of mutations. We've got a lot of genomic data on these, but it's a little bit of a mess, and it's not clean that there's any one repetitive driver between, let's say, even 100 leiomyosarcomas. We certainly know that this is associated with Li-Fraumeni patients. So the TP53 gene, or P53, can be mutated in about 30 to 50% of leiomyosarcomas. In our clinic, one of the biggest sarcoma programs in the world, we see about 1,000 new sarcoma patients a year, even though it's a rare disease. This is often the canary in the mine shaft for a family. If we see a youngish patient with a leiomyosarcoma, we'll immediately get our high-risk genetics program involved, because that may be the patient that ticks off the family to the fact that there may be a germline P53. So we're also seeing this moderately complex, possibly random karyotypic changes and other genomic aberrancies, but nothing driver, nothing really repetitive. So what's the cell of origin? It's obscure still. It's a mesenchymal stem cell almost by definition, and it goes through some level of smooth muscle differentiation. That differentiates it, let's say, from rhabdomyosarcomas, a typically pediatric sarcoma that is striated muscle in the developmental pathway, and where we have, to a great extent, much more successful therapies targeting rhabdomyosarcoma. But leiomyosarcoma, the lyo means smooth muscle, not the type of muscle that you can voluntarily control. This is still a bit of obscure. The interesting thing, it can happen in one of several locations in different patients. The uterus makes up about 40%. It's one of the more common uterine tumors outside of the epithelial uterine tumors, and the uterine leiomyosarcomas are a tad more sensitive to chemotherapy. That's about all we know. That's the level of science. I apologize for this, not my fault. I'm just the messenger here saying that's why we're all here together, trying to improve upon this. There's other leiomyosarcomas that happen in the large vessels, such as the inferior vena cava, the big vein that sits carrying blood from the lower part of your body up to the heart. That's a not uncommon, although all of these are uncommon, part where it can originate, or inside the heart itself. In other soft tissues, very commonplace within my world. And then very rarely there are leiomyosarcomas of the bone, which many of us think probably originate from a blood vessel that's in the bone rather than from a bony precursor itself. Now, one other thing, are there hormonal signalings pathways that are important? There's variable expression, even in the uterine-derived leiomyosarcoma, of the estrogen receptor, ER, or the progesterone receptor. And it's really unclear whether these function. I will note here that even non-small cell lung cancer can have variable expression of estrogen receptor and progesterone receptor. We don't know what this receptor expression really means, but it's something else just to throw out as a clue. What are some unique clinical aspects of this disease, other than it's really not a good disease to have? The major risk is, of course, metastases, as with most cancers. The Ludwig Center here at MIT is very focused on metastasis biology, and the risk of metastases is dependent upon many of the same variables as for other cancers, and certainly for other soft tissue sarcomas. The size of the primary, the depth or location of the primary, and the grade, the mitotic rate of that originating tumor, as well as the presence or absence of necrosis, some of these things that go along with a more aggressive acting tumor. The leiomyosarcomas can recur in the area around the primary surgery, in the locoregional area in that general vicinity, or it may metastasize through the blood, what we call hematogenous spread. This is very unique about sarcomas. They tend not to go through the lymphatics, which is something we associate with, let's say, breast carcinoma or pancreatic carcinoma, where lymphatic spread is common. In sarcomas, they avoid the lymphatics and go through the bloodstream. So, depending on what your spiritual bent is, you could say God is giving us a clue there, or nature is giving us a clue, but there's something interesting about that in the world of mesenchymal neoplasms, that it's hematogenous spread. The main risk, therefore, as it goes back through the blood, where does blood go? It goes back to the heart and gets pumped out for reoxygenation in the lung, and the biggest place we see metastases develop are these micrometastases in the lung that then colonize and show up sometimes a couple of years to several years later, and then after that, the liver. Usually not to bone, and very rarely in my life I've seen tens of thousands of leiomyosarcomas, only seen one or two go to the central nervous system. There's something interesting in that we've never been able to work out. Now, when one takes a survival curve, this is sadly an overall survival curve, relatively recent, 2008, this happens to be the UK, but similar curves are evident all over the place. These are all soft tissue sarcomas once they've metastasized with chemotherapy. If you break out the various subtypes in here, it's not that different for many of the different types other than GIST, where the average survival of GIST patients with targeted therapies is now greater than 7 years. So we've really budged this curve all the way to the right when we understand the disease. With leiomyosarcomas, we haven't. Median overall survival is still 1 year, with some cherry picking in clinical trials, but it's not a good thing once metastases show up. When you break this apart, high grade, meaning the predictive factors of more aggressive behavior, truly do play a role here. Patients with high or intermediate grades die quickly. The low grade tumors do somewhat better, but not significantly a lot. Here's the kind of sad clinical trials we see with empiric chemotherapy, and let me just emphasize that in the world of sarcomas, empiric poisons or cytotoxic chemotherapy was really the gold standard until the late 90s and early 2000s. But this is two different kinds of chemotherapy, simple gemcitabine and docetaxel, either alone with the gemcitabine, a nucleoside analog, or docetaxel, which is a microtubular poison. It doesn't matter if you give them together. Look at the overall survival this is actually just the progression-free survival of a few months. It's not terrific. I say that only because so much of our literature, the clinical literature, is fraught with data like this. It doesn't really help us move forward. What about targeted therapy? It's hard to target something where you don't know where the pathophysiologic driver really is. So the best we see in the targeted therapy world of leiomyosarcomas is a so-called multi-targeted or fairly promiscuous kinase inhibitor, such as pazopanib, which is a very powerful VEGF receptor inhibitor that also inhibits PDGFR, KIT, and a few other things. Pazopanib caused a response in a single leiomyosarcoma patient in Europe, and therefore a big multi-center phase 3 trial was proposed. That's, again, a problem with our world of clinical oncology. But fortunately, it actually made a difference. The pazopanib led to a control of the disease for a grand total of 4.6 months compared to the placebo of 1.6 months. But believe it or not, that was enough to get this drug approved for the use of patients in the United States in leiomyosarcomas and other soft tissue sarcomas. So the bar is set quite low, because the FDA recognizes that patients are in great need of something. And I say that not to be negative, but to actually say I do think our community can do better than this. And here's the latest thing. This is another drug, a marine-derived natural product known as trabectedin, a very interesting DNA minor groove binder that many of us in Boston have been working on for a long time. It's got very interesting biology behind it. And even in this, in a large-scale multi-center study, the best we've done here is to control the disease for an additional 3 months. Even this is under consideration by the FDA for possible approval later on this year. We don't know what their decision's going to be. But honestly, this is the problem with empiric cytotoxic chemotherapy. You don't know how to make it better. You're just basically throwing the dice. If they come out like this, it's good, but it's rarely a big breakthrough. So can we do something better than this? Our colleagues out at Stanford, Matt Van der Rijn in the pathology department, has worked on many ways trying to tease this apart. And several years ago, they really showed that the leiomyosarcomas are the red cluster in the middle. They look quite selective, as you might guess. They're most closely clustering a little bit with the GIST in green. I apologize for those with red-green color blindness in the room. But the bottom line is the leios, as you might expect from an expression array, have certain things that are rather unique. The smooth muscle differentiation program allows them to cluster together. Well, even within this, Matt went deeper and showed that within leiomyosarcomas, there are 3 different clusters of types of leiomyosarcomas. And this has now been even validated further in looking at TCGA, the Cancer Genome Atlas data, showing there really are 3 rather distinct molecular subtypes of leiomyosarcoma. And the gene list has been in these papers, and there's a number of different potential therapeutic pathways. Honestly, it's about 3 levels of difference from really using this well in the clinic. But I think it's a step in the right direction. Let me also add that the guy who was the first author on this paper, Andy Beck, is now in the Dana-Farber Harvard community as a pathologist at the Beth Israel Deaconess Medical Center. So we're kind of importing some talent in this. And I'm just encouraging people to talk to each other and think about how would you hit this? What other things in the research world here that might be in vascular biology, might be in other smooth muscle biology, that could actually help us understand smooth muscle neoplastic behavior? With that, this honestly is simply a list of genes that showed up in those different molecular subtypes. And in 15 minutes, this is about all I wanted to present to you. We've started to understand with the Broad Institute some of the issues around the immunogenicity, or I might add the lack thereof, of this mesenchymal subtype. It's not completely null. We actually have a patient who's had an extraordinary response to a PD-1 inhibitor, but that's only 1 patient out of 50, so you can imagine there's a lot of work going into what made that patient different. So it's not completely negative, but it's a rare event. So with that, I would say this is the note I wanted to end on. New approaches are desperately needed, and that's why we have this bridge project. So there's differential expertise on both sides of town. We have tissues, we have models that are rather proprietary, just like David Weinstock with his leukemia models. We have leiomyosarcoma models from patients. I'm not going to be one of the investigators in this. I'm just trying to stimulate other people to jump into this field, because frankly, part of the problem with rare diseases is you tend to get a universe of investigators who always work together and start to sort of live inside a cloister. And I'd rather open that up, and that's part of the process that Tyler and David and I talked about, trying to use some of the funds to fund this bridge project and open up the universe of investigators here. So that's it.